The Mekong River Basin (MRB) represents a global hotspot of aquatic biodiversity, second only to the Amazon River in terms of total fish species richness. The biodiversity of MRB encompasses over 790 species of primary freshwater fishes, including 28 endemics. The fisheries of MRB is one of the most productive in the world, providing livelihoods for millions. However, the rapid development in Southeast Asia in recent years has caused a lot of damage and stress to the biodiversity of the region. There are many threats to the environmental integrity of the MRB, including hydrological changes from numerous plant damming projects, changes to rainfall inputs, and seawater incursion because of climate change, pollution from urban, industrial and agricultural sources, and changes in flow patterns and sedimentation from land use and agricultural development. All of this can have profound effects on the biodiversity and fishery productivity of aquatic ecosystems and will demand greater management and conservation attention to mitigate these threats. So in order to maintain sustainable development and natural resources, many scientific research projects related to the biodiversity conservation and resource management have been deployed in the area. United States Agencies for International Development (USAID) and the Natural Science Foundation (NSF) funded program Patents Enhance Scientific Research (PEER). Projects are selected according to the criteria of scientific contributions, have an impact on development in the country, and create prospects for strong cooperation between scientists in the developing world with U.S. scientists.
USAID is committed to helping Vietnam transition into a responsible and more inclusive partner. This includes strengthening Vietnam's governance to facilitate broader-based sustainable growth. It also means strengthening the country's capacity to protect and improve the health and well-being of Vietnamese, as well as advancing the U.S.-Vietnam partnership. At USAID, we're always looking for ways to be more efficient and effective in our programming. And this includes supporting research grants like PEER that help us identify smarter and more sustainable ways of doing development. We depend on researchers like the PEER grant recipients in Nha Trang to help us better understand the development challenges on the ground in Vietnam so we can implement more effective projects.
Nha Trang University is currently implementing two projects PEER, Conservation Genetics for Improved the Biodiversity and Resource Management in the Changing Mekong Delta, and Building Genetic Biodiversity Research Networks in the Mekong River. The research projects on the structure and connectivity of populations of fish species with different life cycles and represents the ecosystem zones upstream, estuarial and coastal areas of Mekong Basin, which allows assessment of the effects of changing environmental conditions in the Mekong region by changing hydrological conditions because of the formation of the dam upstream, climate change, and development of agriculture and industry. The project has the participation of partners from Vietnam, Nha Trang University, Cần Thơ University, Cambodia, Inland Fisheries Research and Development Institutes, and Laos, Living Aquatic Resources Research Center. The development of effective strategies for resource conservation and management require, among other things, a firm understanding of population connectivity patterns for the species of interest. Inadequacies in this information can lead to haphazard placement of limited effort and resources meant to assure species persist and available for future human use. Basic genetics and the recently emerging field of next-generation sequencing (NGS) are expanding capabilities for scientific genomics research, with applications to solve pressing problems in biological conservation and resource management. Applications of molecular techniques to genomic questions are limited in MRB, where both biodiversity and threats to biodiversity are most concentrated. Extensive sampling of 3 fisheries representing 3 ecosystem zones, freshwater, brackish and saltwater, was conducted at 14 sites of the Mekong Delta. We expect to observe the 5-scale population structure of 1. Sedentary freshwater species that will likely be influenced by the myriad environmental changes. 2. Estuary species to better understand the influence of salinity changes on biodiversity and fisheries. And 3. Population connectivity changes north and south of the Mekong outflow that will occur due to hydrological changes from damping and climate change. So far, preliminary results show strong population structure from upstream, mainstream, and downstream of the Mekong River. This suggests that localized populations are potentially threatened by environmental changing due to human impact on specific segments of the river tributaries, such as reducing the flow, seawater intrusions caused by dams and climate change. Moreover, there have been changes in the Mekong River flows that could influence separate populations in the north and south. In the process of project implementation, Nha Trang University organized the training workshops on next-generation sequencing (NGS) with the aid of expertise and skills of laboratory professionals from Old Dominion University and Universities of Texas A&M Corpus Christi, USA. Dr. Jonathan Puris and Sharon Furinis will participate in lab work as instructor and teaching bioinformatics. Attending the training were members of the project Building a Research Network of Genetic Biodiversities of the Mekong River at Nha Trang University NTU, Cần Thơ University CTU, Inland Fisheries Research and Development Institute Cambodia, and Living Aquatic Resources Research Center Laos. Together, practitioners, students at Nha Trang Universities and Research Institute for Aquaculture 3.
I think that applying next-gen sequencing to the Mekong Delta is very important. You can learn a lot about it and a lot about the species there and the importance of these species and potential conservation. We use these techniques to gain more information about the genetic composition of these organisms and if they're related to other like species in local areas. For conservation, this can help if you have a species that's depleted from a certain area. You can determine if there's connectivity and if this area becomes protected, if these species can be replaced from other areas. As far as biodiversity, it gives us a better idea of just how different species are from various areas.
That this project will share more experience and to build up the capacity to the young generation staff like me to do more research on molecular research on fishery. I think that I have in my institute have more research that related to the DNA barcoding, and then we will do more research like we need to DNA or molecular method, to do DNA research like to research to discover more fish species in Mekong or such as Tonle Sap River, and we try to continue to do more research on fish in Mekong.
This information can be used as input source for environmental management and resources, establish specific management areas, and develop strategies for biodiversity conservation. These projects will enhance research capabilities in the application of advanced genomics research, especially in the next-generation sequencing to Nha Trang University in particular, and partners in the Mekong River Basin generally.
Thì cái thành tựu của cái dự án này là chúng ta có được những cái phương pháp nghiên cứu hiện đại mà có thể áp dụng để nghiên cứu cái tính đa dạng và bảo tồn cái nguồn lợi sinh vật biển ở Khánh Hòa. Vì như chúng ta đã biết, Khánh Hòa là cái nơi mà nó có cái tính đa dạng sinh vật biển lớn nhất nước. Và với những cái kỹ thuật mà chúng tôi đạt được trong cái dự án mà làm ở sông Mekong, thì chúng tôi sẽ tiếp tục cái hướng nghiên cứu mà ở viện đã tiến hành lâu nay, là liên quan tới cái việc chúng ta tìm cách là bảo tồn cái nguồn lợi sinh vật biển ở tỉnh Khánh Hòa của chúng ta.
Currently, the modern genetic techniques, particularly the application of technology of next-generation sequencing, is widely used in advanced genomic research, contributing to solving many of the problems in agricultural, medicine environment, as well as in the study of biodiversity and natural resource management.
I think the real benefit to the workshop is that getting firsthand experience with data and with training with someone who's developed analysis software really is it's a priceless commodity in terms of how anyone can take that training and apply it anywhere. Like, that's the real beauty of the techniques we're teaching here, is that as long as you have a lab and you have a computer to analyze data, that you can do this, and it doesn't matter where you are in the world. But what matters is that you understand what you're doing, and so that's the real benefit for a training workshop like this, is that young scientists can come and get hands-on experience with people who've developed methods, and so there's no ambiguity. There's no reading from a paper, reading from a blog page in a language that's hard to understand. They can sit there and ask questions and really interact with an instructor, and I think that is something that is truly worth every penny of training costs.
Thì trong quá trình thực hiện dự án thì chúng tôi đã cũng cố gắng hết sức và đã phải trải qua rất nhiều những cái khó khăn. Thứ nhất là những cái kỹ thuật mới, cái kỹ thuật được gọi là dạy tự thế hệ mới. Đây là một cái kỹ thuật rất mới, không chỉ trên thế giới mà ở Việt Nam cũng rất là mới. Bởi vậy cho nên là chúng tôi gặp rất là nhiều khó khăn trong khi triển khai kỹ thuật này. Tuy nhiên thì chúng tôi được sự hỗ trợ rất là nhiệt tình của đối tác Mỹ, là từ trường đại học Old Dominion và hiện nay là trường đại học Texas A&M của Hoa Kỳ đã hỗ trợ chúng tôi rất nhiều trong cái việc là thực tập những cái kỹ thuật, gửi chuyên gia sang hỗ trợ chúng tôi tiến hành những cái nghiên cứu, đồng thời là tổ chức những cái khóa tập huấn cho những nhân viên của dự án, thành viên của dự án để tham gia những cái khóa tập huấn này. Về cái ảnh hưởng của dự án thì dựa trên cái tiêu chí là tiến hành những cái nghiên cứu để có thể là giúp hỗ trợ những cái chiến lược về nghiên cứu về đa dạng sinh học và bảo tồn nguồn lợi thủy sản tự nhiên. Bởi vậy cho nên là cái dự án sau khi kết thúc sẽ có được những cái ảnh hưởng rất là rõ ràng đối với cộng đồng. Trong những cái đối tác của dự án thì ngoài những cái đối tác, những cái viện nghiên cứu ở Lào, Campuchia và hy vọng là Thái Lan và Myanmar, thì chúng tôi cũng có cái đối tác là từ cộng đồng sông Mekong, đó là Mekong River Commission, cũng tham gia trong dự án của chúng tôi. Bởi vậy chúng tôi trong cái tiến trình thực hiện chúng tôi sẽ tổ chức một cái hội thảo, và trong hội thảo đó chúng tôi sẽ mời những cái nhà quản lý, những nhà khoa học, cũng như là những nhà khoa học, cũng như là những cái thành viên của những người mà liên quan đến, có ảnh hưởng đến cái việc xây dựng những cái chính sách hoặc là thực hiện những cái chính sách ở khu vực sông Mekong. Thì chúng tôi sẽ trình bày những kết quả của chúng tôi, và chúng tôi hy vọng rằng là sẽ mang được những cái thông tin đấy đến cho những cộng đồng, cho những nhà quản lý, cho những người trực tiếp thực thi những cái chính sách như vậy.